Okay, let's jump right into this. Welcome back, biology students. I'm acting as your lead biotech lab consultant today for this explainer, where we take these massive, complex scientific concepts and really just break them down into crystal clear, actionable steps. Today, we're exploring literally the architecture of life. We are looking at the foundations of recombinant DNA technology, or rDNA. Honestly, this is one of the most groundbreaking, absolutely fascinating techniques in all of molecular biology. It's the absolute bedrock of modern biotech. If you've ever wondered how scientists are actually able to manipulate the building blocks of life to solve, some of humanity's absolute biggest challenges, well, you're in exactly the right place.
Here's our roadmap for today. We're going to hit the molecular copy-paste, the 5 steps of rDNA, transforming modern medicine, feeding the future, and then we'll wrap up with the recombinant recap.
All right, section 1, the molecular copy-paste. Let's actually understand our DNA. To kick things off, let's establish a really clear biological definition of recombinant DNA, just so we're all starting from the exact same baseline. Recombinant DNA, or rDNA technology, is basically a technique that lets us combine DNA from completely different organisms to create brand new genetic combinations. Really think about that for a second. We're taking the genetic blueprint from organism A and literally splicing it together with the blueprint of organism B. By doing this, we're essentially redesigning the biological instructions from the ground up to create something totally new and highly functional. It's a wildly transformative process that has completely reshaped medicine, agriculture, and the global biotech industry. A great way to think about this is using an analogy we all use every single day, the copy and paste tool. Think about digital text editing. When you're writing a lab report, you routinely cut words from one paragraph and paste them into another to, make a better sentence. Well, genetic editing does the exact same thing, just at a microscopic level. Instead of a computer mouse, we use restriction enzymes. These act as our biological scissors to cut the specific DNA sequence, and instead of hitting Control-V on a keyboard, we use vectors, like plasmids, to paste that specific gene right into another organism's DNA. It is literal biological text editing. Moving on to section 2, the 5 steps of rDNA. This is our core tutorial. So the crucial point you have to understand here is that this isn't random. It's a highly structured, highly controlled recipe. There are 5 deliberate steps in rDNA technology. Step 1 is the isolation of the desired DNA. You have to actually find and extract the exact genetic sequence you're looking for. Step 2 is cutting that DNA using those restriction enzymes we just talked about. Step 3 is the insertion of that DNA into your vectors. Step 4 involves transferring this new combination into the host cells. And finally, step 5 is the cloning and expression of the target gene. This perfectly structured flow takes us all the way from isolating a tiny, invisible fragment of DNA to actually manufacturing a working biological product. Let's zoom in on a couple of these steps, because out in the lab, this is where things get really interesting and sometimes challenging. Step 3 is the vector insertion phase. This involves the insertion of specific genes into vectors, typically plasmids. Why is this so absolutely vital for your notes? Because this is the exact moment the actual recombining happens. The plasmid vector acts as our biological delivery vehicle. By inserting that isolated cut gene into the vector, we're creating the actual recombinant DNA molecule that's going to carry our new instructions forward. You always have to use strict safety protocols when handling these plasmids, but when it aligns perfectly, it's a real game changer. And here's how that builds into the final phases, steps 4 and 5. Once we have our vector loaded up with the new gene, we transfer it into host cells. Think of these host cells as tiny microscopic factories. Once inside, the host cell starts to multiply, which leads to the cloning of our target gene. But actually, scratch that. Cloning alone isn't enough. We need expression. Expression means the host cell actually reads those new DNA instructions and starts physically manufacturing the target protein. That's the finish line. That's how the target gene is successfully transferred and expressed to actually do its job. That brings us to section 3, transforming modern medicine. Let's look at the healthcare breakthroughs. When you look at the real-world applications of rDNA, the medical breakthroughs are just staggering. A primary success story, one you absolutely need to know, is the production of human insulin. By inserting the human insulin gene into host cells, we can safely and reliably produce life-saving insulin for millions of people. No more relying on unpredictable supply chains. It's the exact same principle for vaccines, literally training our immune systems to fight off diseases. Beyond that, rDNA is the bedrock of future breakthroughs in gene therapy, advanced medical research, and even the production of sophisticated diagnostic tools that doctors rely on every single day to catch illnesses early. The advantages here fundamentally disrupt how modern medicine operates. Because we can use host cells as those microscopic factories, we achieve the large-scale production of incredibly useful proteins. We aren't limited by having to extract these proteins from scarce, natural sources anymore. We can just manufacture them safely and efficiently right in the lab. Ultimately, this directly translates to vastly improved disease treatment methods, giving patients much safer, much more targeted, and just far more effective healthcare options. Now for Section 4, Feeding the Future. Agriculture and sustainability. Recombinant DNA isn't just revolutionizing the pharmacy, it is completely transforming the farm. The agricultural applications are massive. This tech allows for the direct development of genetically modified crops. Rather than waiting decades using traditional breeding methods, we can insert specific genes so crops are engineered to resist pests, survive brutal climates, or even carry enhanced nutritional profiles. And these exact same genetic principles are being applied right now to the improvement of animal breeding, creating much hardier, healthier livestock. The crucial takeaway for global markets here is the sheer speed and efficiency this brings to our food supply. By utilizing rDNA, we are seeing heavily enhanced agricultural productivity. Simply put, we can produce a lot more food, much more reliably on the exact same amount of land. It also allows for faster, more efficient biotechnology processes across the board. This is an absolutely critical factor when we talk about environmental sustainability and ensuring global food security for the future. All right, let's bring it all home. Section 5, the recombinant recap, core concepts. If we're going to do a quick mental quiz, the best way to retain all this is to link the microscopic directly to the macroscopic. On one hand, you have those 5 core microscopic steps: isolation, cutting with restriction enzymes, vector insertion, and cloning in the host cells. But remember, those tiny, invisible lab steps equal massive macroscopic impacts. Those molecular copy and paste mechanics directly result in transformed healthcare, scaled-up global food production, and better environmental sustainability. You quite literally cannot have the worldwide impact without mastering those 5 microscopic steps first. To really cement the monumental importance of what we've covered today, I want to read this powerful thought straight from our source material. Quote, "Recombinant DNA technology is one of the most important innovations in modern science, helping researchers solve challenges in healthcare, food production, and environmental sustainability." I think that perfectly hits the nail on the head. It encapsulates exactly why this topic is so foundational to your biology careers and to our modern world. So I want to leave you with a final thought to ponder, and maybe even debate as you continue your studies. How will recombinant DNA continue to solve our future challenges in healthcare and environmental sustainability? We've already seen commercial successes with insulin, rapid vaccines, and high-yield crops. But as this tech grows even faster and more precise, and as we navigate the ethical considerations that come with it, what boundaries are we going to break next? Think about the possibilities. Thank you so much for joining me for this explainer. Keep asking great questions, keep studying those core lab steps, and I'll catch you in the next one.